Welcome everyone. This topic in pathophysiology that we'll cover today is called genetics and developmental disorders. It is one of my favorite branches of biology. I love genetics, and I am not claiming to be an expert. You could spend your life studying genetics and pathways of inheritance, but I find it very interesting. So hopefully this little lecture will provide the same kind of interest to you as it does to me. So let's take a look at exactly what we're talking about with genetic disorders and a little bit of developmental disorders. Prior to viewing this lecture, though, hopefully you've already reviewed some of the background information, some of the background biology on just heredity in general and DNA replication and how we make proteins, and even some on genetic disorders. I've provided this as background information because we don't have the time to hit it as hard as we teach these things. And I mean, you'll hit them in beginning biology, you'll hit them again in molecular and cell biology, and you'll get them in genetics. And I don't have time to review it all, but we'll take a look at some of the basic concepts, and then you will apply these things to some of your analysis as you look at disease and clinical manifestations. Okay, so as a background, these are terms that you should know by now. You're advanced biology students, our nursing majors and MLS students, but two very important terms in genetics are genotype and phenotype. And as you can see here on this part of the screen are Punnett squares. You've sort of done these in genetics and even basic biology, but basically it's a little way to show inheritance from a simple and pure Mendelian genetics perspective. Okay, and if you remember Mendel, he was this scientist that worked with peas and kind of created this two gene, two allele inheritance model. And one of the big part of that is this term right here, or these terms, dominant and recessive, how they're expressed and how much power each allele has. You know your genetic makeup of your biological parents. Sometimes you get a trait that's more your dad's, and sometimes it's more your mom's. And sometimes for a trait to be expressed, it takes two genes for that expression to occur. Typically, that's what we refer to as recessive. It takes both of those. For example, blue eyes and blonde hair are recessive traits, and you need to get an allele from both parents, a gene from both parents. If you have brown eyes and your parents both have blue, there's some explaining to be done there. It's possible, but it's not very common. So again, these terms, genotype refers to the actual genes that were delivered by your biological parents. One allele from your mom, one from your dad. Those would be, we express those in heterozygous, homozygous. These are terms to kind of talk about the genes that we were given. If you're given two of the same, that homo means same, homozygous. So it would be these conditions where you have the two little b's and two big B's. Heterozygous is you got one type of gene, one dominant and one recessive. I don't know yet which one came from your mom or your dad, but you got a mix of dominant and recessive. This one right here, where you have the complete pure Mendelian genetics. If you have a big B and a little b, the dominant one wins out, and that would be expressed as brown in the case of eye color. This would also, when you have two big B's right here, would also be expressed brown. These would be expressed blue, and that's all of these expressions are referred to as the phenotype, in other words, how it looks. Okay, so that was a quick refresher. You'll get that more review of that, but it's pretty important to understand these terms as we talk about genetics. Because, going back to the slide, how genes are inherited will all depend on dominant and recessive. The mode of inheritance is very important, and carriers and their ability to hide dangerous genes in the gene pool. All right, quickly, some genetic disorders. There's basically two kind of ways that we'll look at. You can have a genetic disorder where you have a mutation in the genetic code. Something is wrong with the amino acid sequence when your DNA reproduces, replicates, and transcribes into a protein eventually. If there's something messed up with mutations in those, and we talked about all kinds of different mutations, frame shifts and deletions, and when that genetic code gets messed up, you could have a genetic disorder. A lot of those are metabolic disorders. You see like diabetes and things like that are more metabolic. Sometimes there's serious problems where you have something wrong with the whole chromosome. You could have an extra one, you could have a missing one. I mean, there's chromosomes come in a pair, one side from your dad and one side from your biological mom, your biological parents. And if information is added or deleted or inserted, translocated, it's flopped around. Anytime you mess with chromosomes, it's kind of a scary thought. But you can see different examples here of deletion, and where you have duplication, inversion where things are turned upside down, insertion, translocation. I haven't shown extra ones yet. We'll get to that. But let's say you have a normal set of chromosomes as an organism, and if you get an extra one, something happened during non-disjunction, basically during cell division. That creates something called trisomy or monosomy, which is like Down syndrome is trisomy, where you have an extra one. Anyway, going on, just basically noting that you can have it in the code or in the chromosome itself, and that could lead to problems. A bit of personal information. My daughter, who's now 18, we did amniocentesis during the pregnancy, and so we knew she was a girl, but we also knew that she had what's called a reciprocal translocation in her chromosomes, where basically material from one chromosome exchanged places with another one in a similar fashion, and it was called reciprocal because there wasn't anything added or lost. It was just not in the right spots. It could cause problems because when you break these locations, there might be some gene sequencing that's very important at those locations, but typically the phenotypes are presented normally, and you don't know it because you don't do testing. So there's probably more people in the population that have reciprocal translocations. You just don't know it. All right, I digress. Let's take a look at some genetic inheritance patterns, okay? And this is where it gets a little tricky, but not really, in my opinion. Anytime you see the word autosomal, that means that the pattern of inheritance is not on a sex cell. It's not on one of the sex chromosomes. It's on one of the other chromosomes. Basically, if you're a girl and an X and a Y, those are your sex chromosomes. And there's just one pair. Everything else is autosomal, meaning that they affect the body, not the sex of an organism, and thus meiosis, which is the ability to make gametes, basically egg and sperm. So anyway, autosomal means body, so you could have autosomal dominant, autosomal recessive. Most of the time you hear about a genetic disease that's autosomal recessive, meaning there's carriers out there, they're heterozygotes, and they hook up and they get the disease is actually when you get to two recessive genes, and when you get those, you get the disorder. Sex-linked dominant recessive carry a different, those are on the sex chromosomes, and a lot of times with that you see a sex-linked genetic disorder more commonly in males, because if you remember, you've got the X and then you've got the little, if you imagine this as a Y, those chromosomes, there's a whole part of that Y that's kind of left off and missing an area, missing genes that it could throw a dominant, for example. So it only would take one recessive. Color blindness and hemophilia are examples of these, and it's typically found in males because usually it would take two of those to get the females, and it usually doesn't happen that way. And so there's what we call sex-linked. But we'll take a look at some disorders that are one of the classic autosomal dominant ones, where you just need one gene to get the disease, one dominant gene, and that would be Huntington's disease. And it's a very, very dangerous and lethal disease because it doesn't show up until after you've matured past like 30 to 50 is when it typically shows up, and you've already reproduced, and you might not know it's in your gene pool. Typically, autosomal dominant, especially if you get two dominant lethal genes, usually the baby doesn't even make it. If you get one, usually the offspring doesn't make it past reproductive. They're usually very lethal. But this one, Huntington's, is a little scary because it shows up later. And I happen to know somebody with Huntington's disease. Okay, let's take a look at that autosomal dominant mode of inheritance. When I say mode of inheritance, that's what I'm talking about, whether it's two recessives you need or just one dominant, if it's on the sex chromosome or if it's on the body chromosomes. An autosomal dominant, the mutation is located on the autosomal chromosome, and it's only involving one allele expression of that trait, and like we just talked about, it's Huntington's disease. You can kind of put this together if you look at a pedigree, and you should practice understanding pedigrees because they really tell you about modes of inheritance. It's kind of like working the puzzle backwards. If you didn't have the ability to do some testing, we use these tools all the time before DNA testing was popular and such, but you could work backwards. You could, if you start to map out a person's genetics, like you could find carriers, in other words, because you can see their offspring. You can do this really well with fruit flies and things that reproduce quickly, where you can turn over generations more quickly. In humans, it takes a lot of historical data and information, like you got to talk to your grandma. Did anybody have this? And you can solve a puzzle. It's really cool stuff. I love pedigrees. But you can kind of see, I mean, there's usually keys to pedigrees where this is unaffected, this is affected, females, circles are always females, squares are males. People use different things, and usually there's a little code. But anytime you see a line, that means a mating, doesn't necessarily mean a marriage. So you've got an affected male and an affected female. Well, you can see every one of the kids is going to have it because there's just no chance for the recessives to come into play. And so here you have unaffected, let's see, you have an unaffected female and an unaffected male. They're not showing the disease, but you can see that they get a child with two girls and two guys. The reality is there's a 50-50 chance with autosomal dominance because usually there's one person that's the carrier and they just have that dominant to throw. And so 50% of the time they're gonna, the normal person meaning not expressing Huntington's, would have two recessives because it only takes one. If you had the disease, you'd have that one. So if you drew it all out, it would be like two little b's on one side of the Punnett square and let's do H's. Two little H's on one side and one H on the top and a little H on the bottom. Well, that dominant one is where you get the disorder. So if you do the Punnett square, you would see that 50% of the time that dominant gene will show up in a mating. So anyway, the long and short story with Huntington's is it's about a 50-50 chance if one person has the disease. Typically, again, like the family that I know, it's a very interesting case because the mother has the Huntington's. She didn't show up with it until she was about 40, I think, maybe 45 to 50. Well, her kids were fully adult, like 18 to 24 or something like that, and they've battled with getting to genetic testing. They could find out if they have it or not. But it sort of changes your perspective. Do you have kids yourself? If you know you've got a 50-50 chance here of having the disease, and you don't know it's going to happen until later. Like I said, it manifests later. And so if you have Huntington's in your family, and like your mom has it, and you're a kid, you could choose to be tested, but it might change how you live. It might make you live more cautiously or not have children. So but maybe that's a good thing. So it kind of depends on whether or not you want the information. But it's an interesting case. In my friend's case, the mom has it. The kids, to my knowledge, have not been tested. They do not know yet. There's two kids. And then the interesting thing is the mom who has the disease has an identical twin sister who doesn't. And that's not genetically possible. So what it's telling me is that either that twin it's going to show up later, like in her 60s, or they're not really identical twins, because identical twins come from the same egg and sperm, so the genetics are the same. It's just the expressions will be a little different because it's based on, we'll talk about it later, it's epigenetics. It's your expression of genes has a lot of influences from the environment. But anyway, I'll leave that for now. Moving on to autosomal recessive, most of the time, this is what we're talking about when we talk about genetic disorders. It's the mutation is located on the autosome chromosome again, and it involves two recessive alleles. And it's often involving a character, and you can see the character is usually a character. The carrier is represented by this little line here, where it's a half thing. So you can see this is when the puzzles become fun, because you almost, if you show up with somebody that's affected here from a recessive trait, they had to get one from both parents. So if the parents do not express the condition, they must be carriers, and that's what's represented up here by their parents. And so these are just all the different genetic combinations from these characters, meaning heterozygous, homozygous for homozygous dominant, homozygous recessive, and that sort of thing. It's fun. You can put the genotypes on here if you really get playing with it and can figure it out. It's a good puzzle. All right, looking at sex-linked traits, the mutations, a similar concept, except the traits are found. The mutations are found on the sex chromosomes. Again, as I've already explained, males are more likely to be affected because they don't have that other part to basically they're often sometimes just getting one allele from their mom. They don't have the corresponding one from their dad because the Y chromosome is sort of short-changed. Sorry, males, but anyway. So take a look at sex-linked traits. In again, classic one is hemophilia and colorblindness. My dad's colorblind. I'm not. I'm probably a carrier, or I could be a carrier. All right. A lot of times the inheritance of traits is multifactorial, meaning there's a lot of different alleles that play into the effect of a phenotype. Your eye color really is a multiple allele situation. You also have environmental influences, and these patterns are harder to predict. Some genes are located close together in the chromosomes and making them kind of, they're grouped together so they're expressed similarly, or they have an impact on certain conditions. And so the location on the chromosome can have an effect on the phenotype, basically. All right, that's kind of one part of this, but the other part is just chromosomes. And one of the coolest things that you do, and you still kind of do this one by paper, is you can do this. You can get a kind of a picture of your chromosomes in your mouth, and what you do is you lay them out. You put them together. You kind of line them up based on banding of the chromosomes, and you can use a cheat sheet too, but you in centromeres, and you basically plot them all out, and you're looking to match them up with their pairs when you do what's called a karyotype. And it's basically a map of your chromosomes. So this is chromosome pair number 1, 2, 3, and these are your sex ones, X and Y. See this little Y? This is the short. This is like all these things are unaccounted for on the Y. You can't match up real well. So that's why these sex-linked characteristics are important, why they're more commonly, on males. Anyway, these are the chromosomes. If you would see number 21, if you get 3 of these, that's Down syndrome or trisomy. And basically that's a problem here in the next slide. So if you had somebody's chromosomes and you took pictures of them, you blew them up from a microscope and took pictures, and you mapped them, you found them, you laid them all together, and you found 3 of these number 21s, you would have a condition, a genetic condition called trisomy, or normal, like I said, you get 2, but if you actually end up with 3, and that's the egg that fertilized, assuming this is a female, it doesn't really matter. It says, anyway, it could happen in male or female parts, but if you get this one fertilizing, then you're going to have an extra pair. An extra chromosome, and extra is not good, and missing stuff is not good. It's hard to say which is worse. You almost got some lethal. You got a lot of genetic. You get a lot more dosage when you have 3. It doesn't just one go away. In fact, even as a normal part of your body process at reproduction and fertilization is what we call X inactivation, where a girl is made up of 2 X chromosomes. One of them becomes inactivated early on because you don't want that double dose of things. And so that's how the next topic, this mosaicism, comes about. But because, for us, females are kind of by default a little bit of a mosaic, and it's very interesting genetics. But because one of those X's will get deactivated in a cell, but it doesn't happen immediately. It happens, early on in cell division, but not the first cell division. So you have a variety of traits being expressed. Sometimes it's one X, sometimes it's the other X. So interesting stuff. But anyway, you want to. And if not, you got monosomy or trisomy, and it's not a good situation. Classic one for trisomy is Down syndrome. Okay, looking at these alterations in chromosome structure, we've already talked a little bit about translocation, where one chromosome material changes places with another. Sometimes that's adding or deleting, information gets lost or added in the process. Sometimes it's reciprocal, like in the case of my daughter. There's 2 other alterations in chromosome structure to talk about, and they're confusing terms, but there's a difference. The first one is chimerism. relatively rare disorder, although I did a big study on this for one of my master degrees on chimerism and how it plays a part in determining the sex of an organism from like if you were trying to test like an Olympic athlete to see what sex they were, whether they're gender, male or female. Anyway, you can actually have your DNA show up that with more than one set of DNA in your body, and this happens more than you think. Like for example, in a bone marrow donor thing, you're getting somebody else's cell, so you're going to have a different set of DNA for that cell. And so it's basically a chimerism is a blending of in an individual, but you basically have more than one set of DNA. There's also very common chimerisms between mothers and sons as well, like during the placental, that's a typo there with the son one, but the mother's cells and the son's cells become blended, mostly the direction toward the son, the offspring, during as part of gestation, and so there's maybe a mix of DNA in there. So if you were to do a DNA analysis and pull a sample of DNA from hair or something, you might have gotten pulled the cells that had the XY, or you might pull one that had the XX, and so identifying that gender might become difficult depending on if they were chimera or not. Mosaicism, it's very fascinating stuff. Read more about it. I just love this stuff. Okay, mosaicism is a little different. It's truly more of a blending where you have different genotypes, and you can see both of these. The calico cat is a classic example of a mosaic, and it's a result of that X inactivation. And all that is she's got one X that's throwing some color for white. She's got it might throw some color for orange and black, and it just depends on where it's at. That's what makes her a mosaic. It's not common in humans. Usually you don't have a blue eye and a brown eye, but it can happen. That's another example or phenotype of mosaicism. It's basically when you kind of have your genetics are a little bit messed up, and a lot of this is around X inactivation. But anyway, let's go on. This is getting kind of winded because I like this stuff. All right, what does epigenetic modification mean? I'm not going to talk in detail, but this is a great term. And when I was younger studying genetics, we didn't have a term, epigenetics, or we didn't talk about it much. We did talk about it some, but it's getting a lot more heat and discussion. And basically, epigenetics is the study of cellular and physiological trait variations that are not caused by changes in the DNA sequence. So everything might be fine in your code of your amino acid sequences and your proteins, but there's all this code that you have DNA. Most of your DNA is non-coding. I mean, when I say coding, I mean it's going to tell your body to make a certain protein. And everything is proteins. I mean, all your eye color, skin color, how your pancreas works, all that is based on proteins. And so you have a code for your proteins, and your body's spitting them out all the time. But and sometimes, I mean, not sometimes, also in your DNA are these regions of non-coding. And that's where a lot of your signaling is done, and it tells you how to turn genes on or off and how cells read genes. It's kind of like the programming, the computer program, if you will. It's not a good analogy, but we used to call it junk DNA and didn't think it had much impact. But it's cool in two ways. These non-coding regions are actually highly variable and unique. That's how we do DNA fingerprinting, for the record, when you identify in forensics who did it, kind of thing. And also it gives us a lot more, this is why identical twins are not the same, because there's going to be environmental factors that turn on genes and off and express or impact how they're expressed, how they penetrate, and that sort of thing. So there's a couple examples of different epigenetic modifications, but we're not going to go into, basically it's a little bit of the biochemistry of the gene here. But let's take a quick look at developmental disorders, and then I'll end this long one. But sometimes things go wrong during gestation or developmental processes, and you have some disorders. Just kind of pay attention to those congenital. I don't really talk a lot about them in this chapter, but how do you identify genetic alterations? Basically, you can do prenatal screening or postnatal screening and genetic counseling. All of these things will help. Usually there's I think a rule of thumb: if there's two things that look genetically suspicious, your doctor would probably recommend screening postnatal. Prenatal, you can do it if you are of certain ethnic groups that know of certain genetic disorders that run in your family, not just family history, but your ethnicity. Tay-Sachs is typically found in Jewish populations and such. And so, and sometimes you're just at a higher risk you just know something about your family. If you knew you had, if I knew that, Huntington's, I'm a carrier, then I might do screening and that sort of thing. Sometimes you just do it because you're older and they're just looking to look for Down syndromes. And there's other tests that can help take you a step further, whether or not you need amniocentesis. They can tell things from the blood as well. All right, I think we're getting close to the end here. This is just a map to review some of the things we talked about, different, how the environment will have an effect on chromosomes and genes. It doesn't take much to mess your chromosomes up in the sun, honestly, folks. Use sunscreen. Here's talks about the different things that we've discussed with chromosomes and what can go wrong, and same things with different single gene traits or those mutations. And anyway, I'll let you review that. And finally, these are the cases that you're going to be looking at in the context of genetic disorders. Sickle cell anemia, Huntington's disease, MELAS, Down syndrome. Turner syndrome is basically a female, but it's a weird, you just get one X instead of two. You can have X, Y, Y. All these trisomies. There's Klinefelter's is kind of another one that's similar. There's Fragile X syndrome. These are things that you're going to explore that are related to genetics. All right, thanks.